Welcome to Wave Club, everybody. I'm here to introduce us. My name is Josh Jacobs, along with my partners in Traveling Wave Crime, Bart Ehrmantraut, Uma Mohan, and Irfan Zabet. Thank you all for coming. I'm really happy to have an excuse to focus on science for 1 hour, given the craziness in the world here. Our presenter, so as you probably know, Wave Club is a once-a-month event that we have here to discuss traveling waves in the brain of any species, including computational models. Our presenter today is Dr. Jon Kleen from UCSF. Jon is going to tell us about his group's work studying a spectrum of human hippocampal traveling waves with microgrid arrays. I'm really excited about it because I know a little bit about the work, and it's a different kind of electrode array in a different setting from where traveling waves are typically studied. Jon Kleen is a neurologist and a neuroscientist at UCSF who specializes in epilepsy care. His work combines clinical expertise with research in brainwave analysis, brain stimulation, and memory, with the goal of improving outcomes in patients with drug-resistant epilepsy. He has both an MD and a PhD from Dartmouth, and he completed his neurology residency and epilepsy fellowship at UCSF with Eddie Chang. Outside the lab, he does fun things such as biking in Golden Gate Park and sometimes even hiking in Norway. His background photo there behind him is Norway. I'm really excited to have you here, Jon. Thanks a lot for coming. And just a couple of groundkeeping things for Wave Club in general. The format for this is that we will have Jon present for about 45 minutes. If you have questions, it would be great if you can post your questions in the Q&A feature on there in Zoom. That will help us stay organized. And then at the end of this session, we'll have everybody who posted a question come up online, and if they're comfortable having a conversation and asking about the question interactively, because that's more fun, because Zoom lets us do that. Our presenter next week is Michael Breakspear from the Hunter Medical Research Institute and the University of Newcastle in Australia. And if you are interested in checking out our past or current Wave Club talks, you can check them out on our YouTube channel, youtube.com/@WaveClubSeminar. And we also advertise on both Twitter and Blue Sky, so you can check that out to learn about new Wave Club events. And I think that's pretty much what I wanted to tell you. And then lastly, if people are interested in presenting at a future Wave Club, we're always interested in learning from new people. Well, number one is open. The Wave Club is open to everybody, so please just come and send your name to our email list and sign up. It's a very open and democratic sort of event. Please just join us, open to everybody. And if you're interested in presenting, send an email to me or Irfan or Uma or Bard and let us know. And because we're always looking for new speakers, both nominations for other people as well as self-nominations. So that's really fun. Okay, I'm going to stop my screen sharing and turn the floor over to Jon Kleen here. Thanks again for coming, Jon.
Yeah, thanks a lot for having me. This is very exciting. I'm just so glad that this exists because traveling waves is such an important part of neurophysiology that we're only like scratching the surface of, as you guys know. And a lot of the stuff that I've done is inspired literally by people on this call, a lot of people, especially the Jacobs Lab. So yeah, let me just share here and show those waves to which Josh was referring. And so, yeah, this is recorded, or this was taken in Ålesund, Norway. It sounds like Josh was just there a couple weeks ago. And so, yeah, I took this like 20 years ago when I was backpacking around there. They have beautiful waves in Norway, but then just to kind of imagine if we were to put a bunch of buoys spread out in a grid across the surface, we'd see them bobbing up and down. And if all we could see was the buoys, that'd be like what we see for these electrode recordings, right? But as we can see here, the richness of the spatial 2D pattern is incredible. So there's a lot of detail that we're missing in our recordings, especially if we have sparse recordings. So I'm going to try to talk about what we used in this study, which is microgrid array recording, so 2 mm spacing. I'll dive into that a little bit soon, but yeah, no disclosures, funding from NIH and NSF. So the hippocampus, my favorite structure, probably maybe others on the call as well. It's crucial for memory. It's complex, curved, and deep down in the brain, though. So in rats, it's sort of this dorsal ventral curvature. And then in humans, it kind of like rotated anteriorly a bit into the temporal lobe. But structurally, connectivity-wise, it's roughly still the same. Uh, and there's a lot of different functional organization as you go along the axis of the hippocampus. So it's really important to note that it's not just a homogeneous structure. It's fascinating because it's one of the only structures in the human brain with three layers left, that and the piriform cortex. But the connectivity of it is really incredible. If you go in the dorsal area, you're going to get more spatial learning and memory. Um, Connection or functions relative to the cortical connections, and then the ventral side, at least in rats here, and anterior in humans would be more emotional, social regulation, things like this. But as you can see, there's a list of different structures that connect to different portions of the hippocampus. So along its axis, there's a lot of different connectivity, cortical hippocampal connectivity. And so the question we have for a lot of this work is, how does intrinsic activity flow across the structure, or does it at all? Is it all one structure for a reason? Why aren't all these structures connected along the entire axis? So this is just an image I got from a paper up there, as you can see, Hong et al. Cortical hippocampal connectivity. These are just sort of the best they could do in terms of showing. Well, it's a great job, it's fantastic, but like this just only scratches the surface probably of the different types of connectivity that are set with the hippocampus. It doesn't even really show the connections with the entorhinal cortex and the perihippocampal gyrus or perihippocampal cortex, which are the main inputs in the hippocampus. So there's a ton of connectivity all over the brain, basically, between the cortex and the hippocampus, and it's different along the axis as well, as you can see here. So let's dive into that a little bit, because if activity is to propagate along that axis, it would help kind of sync those inputs, potentially syncing things that are coming from the dorsal or the posterior region to the anterior region and such. But we couldn't really study this for a long time, and we only had a few sparse depth electrodes in humans. And so as this great study in, I think it was 2008, actually, I might have misquoted that, but by Lubinoff and Siapas in rats, they basically put this grid of depth electrodes down into the dorsal hippocampus and looked at the activity. And it seems like one of the first things they noticed was that the phase of theta waves, these are all theta waves, the phase changed consistently as you went from electrode to electrode. And that's suggestive of traveling waves. And you can really see this sort of diagonal appearance here on these consecutive electrodes. So that was the first study to really kind of definitively show hippocampal traveling waves, although it had been suggested in prior abstracts in SFN and such. But they only really were able to measure from the dorsal hippocampus, maybe into the intermediate. And then this study by Patel et al. In 2012 followed that up by recording from multiple sites. This is just one of the rats that had dorsal and ventral hippocampal electrodes, but they also had many other samples. I'll show some more examples of that a little bit later in the talk. But they were able to look at traveling theta waves as well and verified that. And then in humans, we have got this great paper by the Jacobs Lab, 2015, looking with human depth electrodes that were implanted occipitotemporally. So from the occipital, basically angle directly to basically around or right through the long axis of the hippocampus. Now, as you can see here, there's some phase offsets as you go from electrode to electrode, and that's again consistent with traveling waves. So this is really encouraging that there were these traveling properties in humans as well as in rats. And so one caveat to this is that it's a little difficult to reconstruct what the waves are actually doing if you're not able to account for exactly where they are in the hippocampus. And that's the trouble with imaging now. It's incredible how far we've come, but we still are estimating exactly where our depth electrodes are. So this is an example from our center of occipitotemporal depth approach. So you're sampling from several sites in the hippocampus, but they've got variable depth through that structure. And as we know, there's multiple subfields, the CA1, CA3, dentate gyrus. And if you were to do this orthogonal approach, so perpendicularly, we've got two depth electrodes in here, and they're sampling from, sure, the maybe intermediate and anterior, but to sort of try to reconstruct a traveling wave consistently is very difficult with such sparse coverage. And just to give another reason why that is, this is just a cross-section of a rat hippocampus. So you can see the CA1 here. This is the pyramidal layer, this thick layer here, corresponds to this section here, the stratum pyramidal. And then the stratum oriens is above that. The stratum radiatum is below the pyramidal. And if we were to look at the Lubinoff and Siapas paper, basically, when you put a depth down, you sample from multiple sites across these layers in the CA1, just the CA1. You can see that in the parameter layer, which is right about here, can everyone, hopefully people can see my mouse. I'll assume so, unless I hear otherwise. But the parameter layer is right here. And as you can see above the parameter layer, basically the phase offset is relatively consistent. There's no phase offset, and that's quantified here by that paper, just read as a phase deviation compared to the topmost electrode. But then as soon as you pass that parameter layer, you start to get this progressive phase offset. And it goes to about 180 degrees as you get down through the stratum radiatum. So if you have depth electrodes like here in humans, and you're not exactly sure where they're sampling, it's a little tough to make those conclusions. It's a little tough to conclude that they're all in the CA1, for example, in the stratum oriens, when you know that potentially some of them might be in the dentate, et cetera. So that's a huge challenge. And so one way potentially around this, though, is to try to do multiple electrodes with high density sampling, and like they did in the rat studies, as I showed back here. But to do that again with humans is extremely difficult and requires all sorts of IRBs and such. So what we did instead, in this study, I'm going to talk. These two studies that I'm going to talk about is we use this, microgrid array, and it's, as you can see, it's about the size of a dime, 2 millimeter spacing. This is manufactured by Lawrence Livermore National Laboratories, some collaborators we work with, and this is Eddie Chang. He's my main collaborator and also a mentor in my postdoc. And he'd been doing recordings with these on the cortical surface. But then when he was in the OR, he just had this idea, as he does this standard anterior temporal lobe resection, which is the most common epilepsy surgery we do, or he does, I should say. I don't do the surgeries myself. I'm just there. Basically, he resects this lateral temporal cortex for this, again, typical epilepsy surgery. And then in the midst of that, he gets into the lateral ventricle, and then the hippocampal surface is just basically right there. So it's an opportunity to put a recording array down directly over that hippocampal surface. And that surface is actually just predominantly CA1. As you can see here, if we rotate it out a little bit laterally, the surface where he placed this grid is just CA1 adjacent, so right over that surface of the CA1. And as we saw on this previous slide here, if you're above the CA1 directly out to the surface of the hippocampus, which is right here, it's basically phase equivalent with what you would see in the parameter layer, which is a nice consistency that we can use to our advantage, rather than having to worry that we had passed into the stratum radiatum, where our phase offset can be a lot different. The point of talking about phase so much is this is the bulk of how we estimate traveling waves, is looking at the phase. So when we did this in 6 patients, so 2 were awake and 4 were under anesthesia. So the 2 awake patients were both left-sided. Both of them were able to do some behavioral tasks while we did these recordings. And again, this is just in the OR, and those, in one of the patients, the hippocampus was subsequently resected, one of the awake patients. In the other awake patient, it was not resected because that patient just had a small circumscribed tumor in the anterior portion of the left MTG. So it was the same approach, and the same approach in the ability to record from the hippocampal surface, but without the need to remove the hippocampus, because that patient did not have epilepsy. They basically had presumably a completely normal hippocampus. And that was a very, very just stars aligned sort of fortunate data set, because that patient also did the most behavioral trials that we had. So when I took these recordings and just looked at them, one of the first things I wanted to do was filter in the theta band and look at to see if there were any phase offsets. And sure enough, there were a lot of phase offsets that were consistent. As you can see here, it basically looks like this phase offset from electrode to electrode over time, which is typical or suggestive of traveling waves. And so, and just plotting this same data as like this LFP data as color, we can just see this nice diagonal here that is suggestive of traveling waves. But the thing I noticed, like right off the bat, was if I looked a little bit later, it was the opposite. The diagonal like shifted, and then it would shift back to this sort of other diagonal, and then back and forth. So I already was a little bit, very skeptical initially that this can't be right, because all the data so far had been one directional. So in both animal studies and in the human study, it had all shown traveling waves traveling toward the anterior portion of the hippocampus, or the temporal traveling. So this would suggest bidirectional traveling. So I wanted to dive into this a bit more, and I took basically the very similar, if not almost identical, approach as the Jacobs Lab did for their 2018 paper in cortical traveling waves, and got the power spectrum from each of these electrodes over the course of the whole recording, and then just fit them to just a linear fit, and just did this sort of 1/f adjustment where I could pick out the peaks. And I found a couple of peaks for most patients, actually 2 to 3 peaks. One patient, I think, had one peak. But for the most part, just using these same identified criteria as that cortical traveling wave paper, I found multiple peaks in many of these patients' electrodes. And then this just shows you the composition of all patients. So, sorry, there's not 6 under anesthesia, there's 4, my bad. So there's the 4 solid lines are the patients under anesthesia, and the 2 dotted lines are the awake patients. And so when they were under anesthesia, there was a lot more of these theta frequencies we saw in these recordings, versus when they were awake, there was a lot more sort of like delta theta junction range, and then higher up here in the alpha range frequencies. And then not too much once you get past 15 hertz. So we kind of limited the study to 1 to 15 hertz. Um, but evaluating wave phase, and I don't know how deep I need to get in the details, but I thought I'd just kind of bring it back to basics here quick. When we talk about wave phase, we talk about it in degrees because it's just like a circle, right? It's a cyclical data. It's circular data, I should say. So 0, 90, 180, 270, and then beginning again. It's just like waves in the water. So when we analyze traveling waves, or when others analyze traveling waves in the water, it's the same idea. You can look at all the similar parameters that we look at with LFP voltage. So here's just an example of raw signal, and then theta filtered signal, and then the phase of theta. So it just goes 0 to 360 and then returns again. Anyway, not to get too deep in the basics, but I just want to make sure if there's anybody that's kind of new to this, that they have an idea what we mean when we talk about phase. So here's just a full cycle of a sine wave. And then if we were to take the phase on each electrode at a given time, we could plot it as a color here. And that's what this is. So this is electrode number 1, number 8 over here, and then number 32. So basically 4 rows by 8 columns. So that's our 32 channel recording array. And this is phase plotted at a particular moment. So we can sort of see this gradient here, but to show that a little bit better, oh, and then there's the superimposition. If we wanted to figure out if this wave was traveling, we could plot this model. So this was basically the x position and the y prediction, the y position predicting the phase. So it's a 2-dimensional regression model. A plane wave model is another way we refer to this. And so the gray plank there is just a null model. So that would assume that there's no phase offset. That's if it was just kind of random noise, we would expect it to be flat. But if it was traveling, we would expect there to be some sort of an angle, which there is here, as you can see. And then we fit the plane wave model to that, and that's what this grid sort of structure looks like. And then the residuals, the actual phases, are the black dots with lines connected to show basically how big the residuals are for each electrode at each point. So now that we hopefully have understood what we're doing with that phase wave regression, or that plane wave regression, let's just take a look at that recording that I showed you a little bit ago. So we see these phase offsets here in this direction. We see these phase offsets over here in this direction. But let's just show this as a movie. So we have phase down here, and then we have the analytic amplitude of those same waves. And this is for a wave that was, or an oscillation that was 13.8 is a peak frequency in this particular patient. And that's filtered with a 1.5 hertz, or so basically 3 hertz bandpass around that peak. So if I play this, as it plays, you're going to see the wave direction shown here. And then you're also going to see the R squared for this plane wave model, just shown at the bar here. So of course, higher is better. 1 is perfect fit, and 0 is like a null fit. And as it gets closer to 0, I basically make this a lot more transparent, just to reflect that that's not like a really consistent traveling wave at that time. So if I play this, basically, if you watch the phase, that's kind of the main thing to show you. This is, again, filtered data, but I haven't done really anything else to this besides downsample before. And you can see that clearly. And then now this sort of breaks down. There's no longer like a consistent traveling wave. And then as it resurfaces and gets that structure again, that spatiotemporal structure, you can see that the direction of the wave on the phase plot over here is now the opposite direction. So at first it was traveling toward the temporal pole, now it's traveling toward the septal pole. So, happy to show that again later too if people want. But so I was very surprised by this because this suggested traveling waves in the hippocampus were bidirectional. And I was basically seeing this on almost all oscillations that we had. But to just, again, make sure that we weren't seeing some kind of noise, maybe just some sort of artifact from another system, we did a couple of controls here. So this is, just in the supplement of a paper that we put out, and it's a manual rotation thing. I actually want to put this in the main article, but there just wasn't enough room. But I thought this was really interesting, and I hadn't seen this done before, where Eddie had put the grid on the hippocampal surface, which is right here in the background here. And then we recorded it for a few minutes or so, like several minutes, I think. And then we paused the recording, and Eddie rotated it about 25 degrees or so. This is the approximation. This is going off the video picture. I tried to estimate as best as I could. And then we recorded again, and I found the same peak frequency in both of those recordings, and so I fit it to this, and then we can basically see that despite a counterclockwise rotation, we had a clockwise rotation, and we had a counterclockwise shift in the mean angle of these traveling waves. So to me, this suggests that this is a biological property of the tissue, that we just rotated the mechanics of our electrodes, and we had a corresponding counter rotation of the perceived angle of the wave. So again, I feel like this is consistent with a physical property of the hippocampus where these waves are flowing. Now, another control we did, which just kind of came up in the reviews when we were in the reviews for this paper, and I ended up being really happy that we did this because it gave a lot more credibility, I think, to looking at this. So basically, with this rectangular grid, if we were to do a null model, which is where we randomly shuffle the electrode numbers, therefore the locations of it, and fit the same plane wave, we would get these gray traces. So this is a 9.2 hertz wave here, and this is the gray distribute. The gray is the chance distribution, and I did that 10 times with shuffled locations of the electrodes. So you can see it's not a perfect circle, it's sort of like an oval. So when we have a rectangular grid, the chance distribution comes out an oval, and that's just a property of the regression and how we're doing it and where we have data points. So the key part is, are the actual angles outside of that distribution. So the green shows the actual angles of that, and so certainly they're well out of this distribution. They're bidirectional, so this is toward participant one had a right-sided grid, and so this is anterior-inferior, and then this would be superior-posterior, but certainly outside of the chance distribution here. And then we can see across all the frequencies for that patient that they were bidirectional, mostly toward the temporal pole in that patient. And then participant 5 over here is a left-sided patient. Same idea. You had this sort of oval-looking chance distribution, and you had these bidirectional traveling waves outside of that distribution. And then I showed just basically we do the same thing. This is where the control part comes in. If we're getting a different chance distribution for a rectangular fit, well, what happens when we take a square? So we just took the middle square of the electrode, so 4 by 4, and there we get a nice circular distribution. So it's sort of balanced in space, or spatially balanced with that 4 by 4 grid. But again, the same angles come out for the actual data. So again, this is just done using these black electrodes here, and we get a very similar distribution of the traveling wave angles. There's also some electrodes that are missing here too, these ones with the hatched out, and that comes into play later. But basically, the control was trying a bunch of different shapes and constellations of these electrodes here. So now we have an empty square, we have like a stop sign, we have an empty stop sign. And as you can see, the distribution of chance distribution gets a little bit funky. It gets kind of quite interesting depending on where we have electrodes sampling. And then we did this with even more experiments. So we had like a long 2 by 4 grid. We had like rotate that 90 degrees. We had like a 4 by 2. And then we had this sort of like 3 by 2 sort of diagonal version as well that we did in a couple different ways. So if we take all these and overlay them, basically the distribution, it's different slightly because of the estimation. We're always going to have some noise, but it's basically the same in terms of bidirectional traveling waves. And so that again spoke to me that these are properties of the tissue itself that are coming through, and not just due to the recording array and artifact. So so to make sure that we were looking at traveling waves in the first place, we took the same approach as the Jon et al. Paper from 2018, which is we did this sort of chance distribution where we randomly shuffle the electrode locations, and then we see what the R squared is. And then we see if the actual was beyond the 99% confidence interval, which it was here. And so again, like these chance distributions, or these chance R squareds, those are the same models that we use for these chance distributions here. I guess I should say this one, which is the full grid. And so this is just one single time point in that data set. And if we plot all the directions of all the valid time points, meaning all the time points in which the actual value was above the 99% confidence interval, we get this distribution of bidirectional traveling waves. And that's just for one patient's at a single frequency. But if we take all the patient's directions at all their different frequencies and we plot them overlaid, we can see that it's basically quite consistent between patients. The right-sided patients who were all under anesthesia, four of them, we had the angles in the posterior superior direction and the anterior inferior. And then on the awake left-sided patients, there were two of them, it was basically the mirror image, as we would expect if this was a consistent property of the hippocampus. And I show these angles kind of oblique, sort of not oblique, but diagonal, because these are the directions, right? But I kind of wondered, like, why is this a little bit offset? It doesn't exactly follow the longitudinal axis quite. I thought that was a little bit strange. And so this is another control that we did. Again, this is embedded in the supplement, but this is one of my favorite analyses that I think I've done in my career. Really, is looking at this. So we took the grid, and instead of taking the full grid, we took a 4x4, the anterior-most 4x4, and then we did the distribution and also got the shuffled version to make sure it wasn't beyond, or that it was beyond chance. And we took that same 4x4 and just shifted it back by 2 millimeters. And then we did the same thing again and again and again. We went from the anterior-most to the posterior-most data points, and we got a nice distribution that was beyond chance for each of these. But you might not, I don't know if it's quite clear on these different shifts. So if I overlay them, it might be a little bit more clear that as we went in more posteriorly, actually, so this is a different one. As we went more anteriorly, there was a counterclockwise shift. And another way to say that is basically if we overlay these in relative proportion, relative position every 2 millimeters, we're getting a flatter distribution as you go forward. And what that suggests is that if we record from the more posterior section, it's got this upward diagonal angle. If it's more anterior, it's more of a flat. And that basically just corresponds to the hippocampal curvature. So the reason I think that it was sort of a diagonal when we use the whole grid is that actually if we look in little pieces of the little parts of the grid, it is actually curved, and it's curving along the length of this longitudinal grid, this 4x8. So that was another nice control, but also another nice verification that this is following the hippocampal structure itself. And if we overlay, again, all these patients' traveling waves, these are all the peak frequencies that we got, so up to 3 per patient from the top to bottom. And then one patient only had one frequency that came out there. But basically, they are all quite consistent with this bidirectional traveling waves. But another interesting thing was that the patient that had, so I should say that, 5 out of 6 of these patients had epilepsy to varying degrees. And one of the patients in particular had severely abnormal what we call mesial temporal sclerosis, meaning like hippocampal, sort of like scarring over time, and that's leading to the source of their epilepsy, the source of their seizures. So in this patient, we had 2 peak frequencies, but they were only unidirectional, which I thought was quite interesting. That could suggest, this again, speculation. Don't have proof. We would need a larger study looking at more patients. But to me, it suggested that there may just been some faulty circuitry that kind of broke down in terms of the anterior traveling waves, whereas it was a bit more intact in the patients that had less or no hippocampal disease. So yeah, that was quite interesting. Look at just sort of looking at sort of all time points for baseline traveling wave properties and behavior, properties, but then moving into behavior, again, 2 of these patients actually did a visual naming task. And we know from prior work, by the, a variety of labs, actually, Zagul lab, Jacob's lab, it's Norman did a bunch of work on this. There's a couple other papers showing that when you do a visual naming task, you actually get hippocampal high gamma activity increasing, or just hippocampal activity in general. You can even see sort of like almost like an ERP, like a voltage shift as soon as maybe about a half a second or less before, or once you show an image. So we took the traveling waves, or we took basically the data and aligned it to this picture task. It was just showing a picture and then they would say the answer as soon as they could come up with it after. And we did this time point by time point, locked all of these trials, and we show the angle. So as you can see here, the angles of these traveling waves right before you show the picture are basically bidirectional, but there's no consistent directionality. So the directionality consistency is basically just like it's just the circular mean essentially, and you can use a Raleigh test to test that. But essentially, at each time point, we could test how consistent the waves were moving in a particular direction, or how inconsistent they were. So it's quite inconsistent until about zero point 0.4 seconds after you show the pictures. And if we look way over here at like 0.9 seconds, or almost a second after you show the picture, almost all the trials had a traveling wave moving in the anterior inferior direction, basically toward the temporal pole. And this is again like quite striking to me, at least, where there's only like a handful of of the trials that are moving in the opposite direction. The vast majority are moving consistently toward the temporal pole here. And then if we align it instead to when they speak, so this is speech onset, and look at these again. So basically, right before and around the time of speech, we get the same sort of anterior flow of neural activity. And then shortly after that, it sort of breaks down and they go back into chance. They may be moving toward the temporal or septal pole, or maybe even some other small angles there. So I mentioned sort of like an ERP thing before. So that's when we think about ERPs, those are like delta waves almost, right? They're sort of like low frequency shifts in voltage. And so the concern would be, since this was a 1.9 hertz oscillation, could this just be a transient delta shift from sort of like an ERP thing? So we looked at the analytic amplitude across channels, and we also split this into, I should say, we split this into fast and slow reaction time. And basically, with fast reaction times, we saw stronger directionality consistency at those same times that I showed you before. But getting more into this other control, looking at the analytic amplitude across all channels, the peak in the analytic amplitude was about zero point 0.6 seconds after, which is consistent with prior research. If you show like a picture and you'll see sort of a subtle low frequency shift, but it's very transient. But the key is that the directionality consistency was really after that. So after that peak, it was really just kind of after that these low frequency oscillations stayed consistent and then sort of dropped out again. So I thought that was another nice control. But do they always travel toward the temporal pole? Well, we know that they travel bidirectionally. So is there another behavioral correlate? And at first, I didn't really detect this because I wasn't really interested in the intertrial interval. But when I extended it out and started to look, even before the next trial or after the last one, I saw this interesting shift. So if we look, this is the same sort of data that you saw before, this anterior sort of traveling consistency right after they speak the word, but then they wait for the next trial. And there's a variable delay. We don't always give it at a certain time, but so they don't know when the next trial is coming, but they're anticipating it. And about 2 seconds into that, or just before that, you start to see very strong directionality consistency again, but now this time to the opposite direction, towards the septal pole. So behavioral correlate of this oscillation is that it appears to be traveling anterior inferiorly toward the temporal pole when the patient has seen the image and is trying to come up with the answer. But then when the patient is doing a completely different cognitive function, just waiting for the next trial, they're traveling the opposite direction, up towards the septal pole. And then this is for the 1.9 hertz oscillation. Interestingly, at the same time, same patient, we had a 13.8 hertz oscillation. And that oscillation did not have this consistency around the time of speech, but it did during the intertrial interval. They would sweep up toward the septal pole again. So we have two different frequencies, different, people call like different wavelengths, right? Different types of potential information happening at the same time, and potentially doing like different functions here. So you could these could be different resonant frequencies of the cortical structures with which the hippocampus is interacting. It could be different intrinsic rhythms. There are people probably smarter than me on this call that would know the answers to why we might get that. But it is striking me that same patient, two different frequencies with different relative consistencies of how the waves travel. And that's, to me, again, suggests a mechanistic role of these in cognition. We also looked at the topography of the wave amplitudes by direction. And so just briefly on this, we wanted to see if we can predict which way they were going to flow based on the hippocampal activity. And I thought, based on some prior work, including on the Jacobs lab, actually, that maybe if it was more posterior activity or strength or amplitude of the activity of that oscillation. They may travel one direction versus more anterior strength of the activity may travel the opposite. But that didn't really come out. It may be because we were only sampling from the anterior and the intermediate portions of the hippocampus. So I use this SVM model instead. So it's a simple machine learning model where you feed in the analytic amplitudes at each of these electrodes to try to predict a binary decision of whether it's traveling toward the septal or temporal poles. And that came out very consistently for essentially every traveling oscillation. We could predict which way it was traveling based on the pattern of activity, the topography of activity across the hippocampal surface. So in terms of conclusions on the low frequency oscillations, 1 to 15 hertz, they all spatially temporally seem to organize. They all seem to have traveling properties. It's not just theta, it's delta and alpha as well. And the direction of the wave propagation may be a novel biomarker, potentially for distinctive computations that are happening on sort of mesoscopic scale. And moreover, the hippocampal street apparently appears to be a two-way street instead of a one-way street. That's their conclusion out of this, which may suggest a hierarchical influence on the propagation route. It may relate to the hippocampal or the cortical inputs that are coming, sort of driving different patterns, or the cortical hippocampal interactions. And bidirectionality may have some relevance for memory sequence order. We think about phase precession, but also replay and pre-play, so forward versus reverse sequences. And again, none of this is shown in our paper. This is just sort of speculation about the potential mechanistic roles that bidirectional traveling waves may have. So to kind of keep moving, because I don't want to run out of time, which I often do, I want to definitely leave time for questions. So moving on, that was looking at low frequency waves, but we wanted to also look at higher frequency activity to see if there were any spatial temporal properties. There's been a ton of work on hippocampal ripples in the past multiple 4 decades, I guess. Largely in rats, but in the last, 2 decades, especially the last decade here, it's been just really amping up for human ripples. And so, ripples are these bursts of high gamma activity that happen in the hippocampus. They've also been found in the cortex in ways, but like the hippocampal ripples are a very distinct sort of flavor where they're predominantly CA1. They're driven by CA3 neurons that are interacting and igniting basically a large population of CA1 neurons. And they have roles in memory consolidation. They also, and in humans, have been found to have a role in dynamic memory sort of performance and recall. So this is just to show across species, ripples are seen in the hippocampus. And then this is another screenshot out of that nice paper by Patel et al. In the Bidirectional lab, looking at 10 different sites. And what they did in that paper is they did a theta paper, but then they followed it up with a ripple paper. And these are the 10 different sites as rows. And then these are the different types of ripple behaviors that they saw. So basically, you see this. This is a spectrogram here. So time on the x-axis, frequency on the y-axis, and you can see nice synchronous ripple here. This is a septal-temporal traveling ripple where it moved from the septal area and then progressively, as time went by, got down to the temporal pole. And then the reverse here, they saw bidirectional propagation of ripples essentially. They also saw very local ripples, sort of dual positions of ripples, and then sort of like a reflection of propagation. Very importantly, I've been talking about phase propagation essentially this whole time. This is not phase propagation. This is amplitude propagation or amplitude traveling essentially. So they were watching the peak of where the ripple power was and then tracking its position, the peak activity position. So you're kind of just tracking the most active neural population at a given time for a ripple as it moves from one area to another in the hippocampus. So we definitely analyzed that. We've been doing that more recently with the amplitude tracking. But the bigger question I had off the bat was I want to look at phase tracking because that's, again, following up what we've seen with low frequency waves. And, with this sort of sampling, it's difficult to do phase tracking, especially if it's moving so fast at fast frequency. But if we have evenly spaced electrodes on a grid, we may be able to track these faster frequencies, hopefully with more utility, who knows. And so this is just a raw recording of, here's raw data here, and then you can see this is a pretty okay ripple here. But when you look at this, it sort of catches you off guard because most of these channels aren't involved. But when you look at that, just the unfiltered trace and the filtered trace, you can see that it looks a lot more like a ripple, what we would expect. And so we did a standard ripple detection procedure. We had used a ripple detection procedure from the Norman et al. 2019 paper. And Itzek is actually, Itzek Norman is a postdoc in Eddie's labs who's very helpful in sort of like, the pre-processing and making sure that we're kind of following best practices for this as best we could with ripple detection. So if we look at this, it looks pretty good. But then if we look over here, we see there's only a few channels that have that, the ripple. So that already suggests that they may be pretty confined. And then this is the peak channel, just showing the spectrogram here. Again, the raw trace and then the ripple trace as well are shown there. And this work, just of note, was pretty much all the work, or almost all the work here in the ripple study, was done by Monica Chow, who's an epilepsy fellow, but has essentially been acting as a postdoc in my lab for the last couple of years while she was finishing residency and epilepsy clinical fellowship, which is very, very busy. And so I'm very impressed at what she's been able to do with this project. So, so again, when we detected ripples, just looking at the properties of them, our pre-processing was essentially just downsample the original 24 kilohertz recordings to 512 hertz. Then we did a 70 to 150 hertz filter, got the Hilbert analytic amplitude, and then z-scored it by channel for each recording. And then for the detection, we did a channel by channel detection, which is what many papers do. So every channel we just detected ripples independently. But since we had this sort of spatial this advantage of having a 4x8 grid, we just assumed that they weren't going to happen on just one single channel. And if they did, could it be spurious? So we had another requirement that they had to be on at least 6 channels with a z-score of at least 2.5 above baseline. And then they also, of course, like other studies, had to be ripples less than 20 milliseconds or above 200 milliseconds were discarded not to be ripples. And we merged any that were within 30 seconds of each other. And this is just the grand average of all ripples in the spectrogram here. And then we get the mean here. As you can see, this is all time locked to the phase, the closest phase trough that's nearest to the amplitude peak. So that's why we get this nice little ripple oscillation across the mean here. If we just average them without adjusting, aligning to phase, then this should be practically a flat line. But since we aligned it to the phase trough, that's why we see this nice ripple there. And then this is just the distribution of the number of ripples and where they were detected. So in terms of the peak, so the strongest amplitude peak for any given ripple is just shown here as a number. So the number of ripples, number of ripple peaks that happen at a given electrode. So they kind of happened all over the grid, as you can see. Maybe some areas more than others, more anterior, more posterior here, posterior, superior. It kind of makes sense that not a lot were picked up on here because this electrode is almost off the hippocampus, basically, towards the infero-posterior edge. And then the properties were, peak frequency, we got what might be expected, especially since, a lot of times people might not realize that you're going to get peak frequencies within the ripple range because you're filtering in the ripple range. Of course you are. But it was nice to see that it wasn't all just smushed to the lowest frequency. The peak frequencies was, 80 to 90 hertz or so as expected, as seen in prior papers. And then inter-ripple interval was just sort of this skewed distribution, but it wasn't, completely down to the shortest possible duration. It was sort of flat once we got to these really short durations here. And then the peak z-score was around 5 z-scores above baseline. And then the number of electrodes above threshold tended to be around like 7, 8, 9, 10, maybe somewhere in there. But we certainly had some that were over half the grid. But basically no ripples were the entire grid, which is interesting. And then getting into the spatial idea. So the spatial breadth of the ripples, we wanted to look at from the peak channel. So what was the ripple amplitude in terms of z-scores? And that's shown for patient 1 here in the blue, and then patient 2 in the orange. And so the peak amplitude, as we saw over here, was around 5 hertz, 5 z-scores. But then if we looked at the next electrodes around it, so sort of like a circle around that peak electrode, it dropped down to like 3 z-scores. And then if we look to the next circle, so 2 removed from the peak one, then it dropped down to like just over 1 z-score. So the point of this is to show that it's a pretty rapid drop-off. When you go from the peak, it drops off rapidly in space in terms of how strong that ripple is. Again, speaking to the fact that these ripples seem to be quite local phenomena. That's suggested by the Bijaki work for many of the ripples, but also just very consistent for what we saw here. And again, since we have behavior on these patients, in addition to verifying the properties, we wanted to see, does this actually line up with behavior? These are patients that are undergoing an anterior temporal lobe resection, and some feedback that we've gotten even on grants on this is that this is like a disconnection. So you disconnected the cortex. These whatever you are picking up on might not be normal neurophysiology. Well, to retort that, um, these are the ripple rates that we saw, sort of baseline around zero point 0.2 here, and then as soon as you show the picture, the ripple rates tended to jack up pretty strongly, and that's especially for novel pictures. So when the patient saw the picture the first time, the ripple rate would increase, and this was significant here as a cluster-based permutation test. And then when you showed the same trial again, it picked up again, but in a more blunted form. And then patient 2 here, we didn't have a lot of trials in patient 2. The patient also had interictal spikes, which we had to avoid those trials. So we had a really limited number of trials for that patient, but 43 trials for the patient 1, split into novel and repeated. So I think it was 23 and 20, respectively. And then around the time they speak, it again was like increased here. So this is kind of a short variable time frame between stimulus and speech, but then they would drop off right after speech. And the patient 1 also did about just a handful of, well, not a handful, like I think 6 or 7 recall trials, and just squeaked by in terms of slight significance on free recall, showing like a higher ripple rate. And this, it's just barely significant there, but it's really consistent with what is seen with depth electrodes in the hippocampus. Many people, many labs have shown this sort of ripple rate increasing right before speech. And to kind of dive quickly into the traveling properties of ripples. So again, we didn't look, well, we looked at analytic amplitude, the amplitude peak and how that propagated, but I won't include that here because we're still kind of like processing it a bit and trying to align between the two types. But for phase or cycle by cycle traveling, we definitely saw traveling properties of these ripples as well. So this is a one ripple that seems to be traveling toward the septal pole. And this is another ripple that was traveling toward the temporal pole. So again, here's the fit of the model. Sometimes it was really nice fit, other times not so nice. But again, we did the same approach where we always did a 99% confidence interval of sort of shifted spatial positions. The angles tended to be very consistent with what we saw with low frequency oscillations, except much more septal oriented. So about a 3-to-1 ratio in ripples traveling toward the septal pole compared to the temporal pole. And we tried to split this out in terms of, if we look at all the ripples, it was this gray trace here. And then within task, meaning between the stimulus and speech, it was still mostly septal. And then if we looked at the ripples in the intertrial interval or outside the task, or even before the task started, basically the same distribution. So no real difference between when they're actively doing the task and not actively doing the task. We even split into animate and inanimate, thinking that maybe some would be more close to the anteriormost hippocampus or travel in a certain direction since the amygdala is one side. But still, again, no significant difference in those populations and that behavioral split. In terms of wave properties, so this is sort of the same data we saw before, very similar. But these two plots here, I want to point out, the wavelength was about like 30 millimeters or so. So maybe like half or a little bit more of the hippocampal longitudinal axis. And then the wave speed was very rapid. It was about, 2,500 millimeters per second, so about 2.5 meters per second, which is much faster than the low frequencies that we saw, which were more like, 3 to 30 meters, or 3 to 100, I think, meters per second, or millimeters per second. 30 to 300, excuse me, sorry. And then in terms of traveling properties, whether that had anything to do with behavior, we split this into septal traveling and temporal traveling. And I guess kind of, the point of this is to show that when they were traveling towards the septum, we saw a lot more of those ripples increase right after showing the stimulus, maybe a little bit though for the septal. And so this didn't really come out significant. And we also show the non-traveling ripples here as a proportion. And so not really much difference, except there were definitely a lot more non-traveling ripples during the task. Maybe there's a functional role of non-traveling ripples as well. But then when we looked before speech, sort of again, it kind of thinned out the septal, or the ones traveling toward the temporal pole, and we have a predominant septally traveling population that seemed to be related to behavior. And again, in the interest of time, maybe I should probably wrap up pretty quickly. I'll show one more analysis here. And that is that, I don't know if people have read this Logothetis paper from 2012. It's a beautiful paper. And in it, they talk about ripples. They look at ripples in primates. And Logothetis kind of pointed out that the ripple band overlaps, of course, a lot with the hippocampal, or with the gamma and high gamma bands. And we can see things like theta time-locked gamma and co-modulated gamma looking like ripples sometimes because it's little bursts of gamma. So a way to kind of delineate between ripples and gamma is to what they did in that paper was they used non-negative matrix factorization to just put all of these events detected into a model and sort of have an unsupervised delineation of what came out. And they were able to delineate ripples from high gamma or gamma, and as well as like a HP sigma, which they looked at was below 20 hertz. So it's a way to identify spatial temporal features, but also account for and thus screen out other neural events like high gamma. So we apply this to our grid data in a spatial spectral way. So not only did we have frequency as part of the as one dimension of the data that we fed into the non-negative matrix factorization, but we also had the grid position, so the electrode number. So it's essentially like a spatial spectral analysis. And we widened out our band since we were going to be able to account for gamma in this way to 20 to 200 hertz. And then we loosened up our detection to just 3 channels reaching above 2 z-scores, again, with the idea that hopefully this would help us delineate from high gamma. And we parsed it onto 3 motifs to explain the data. And we had the first 2 motifs came out really pretty consistently with ripples. Their peak amplitude was around 85 hertz or so. That's as you can see here. And this is the grid. This is actually the grid amplitude that you're seeing here. So one population, motif 1, was more anterior position, and then motif 2 was more posterior position. But they both were around the ripple band peak frequency, which we would expect. Whereas the third motif was basically around where we'd expect a broad high gamma signal to be. So this is nice in that we could basically use these metrics of motif contributions to reflect how like events that were more likely to be ripples. So we essentially excluded motif 3, which is thought to be high gamma, and looked at motif 1 versus motif 2 and tried to see if the direction of travel was any different between the two. Basically, again, these motifs explaining ripples that appeared to come from the anterior for motif 1 or posterior for motif 2 positions of the hippocampus. Not really a consistent direction relation, though, for motif 1 versus 2. So that already was suggestive that no matter where these ripples are coming from, they could travel either direction or any direction. And then splitting this into behavior, motif if it was motif 1 was stronger than motif 2 for a given a given event, we looked at those first and we saw that essentially similar patterns where it increased during visual naming maybe a bit. But if we looked at the ripples that were more attuned to motif 2, which is more posterior, meaning more intermediate in the location of the hippocampal body, there's a lot more consistency for visual naming, like especially the novel visual naming, where it jacked up pretty quickly. So more posterior intermediate ripples tended to be more associated with this visual naming stimulus the novel visual naming stimulus. And then not much for before speech, but we did see a little bit more likelihood for them to be involved in recall for these more posterior situated ripples. So conclusions along for the ripple propagation is that, as you can see, text heavy here, sorry, but ripples recorded from the CA1 adjacent surface seem to be consistent with the ripples that we would see from depth recorded ripples. They propagate towards the septal pole mostly, so a 3-to-1 ratio compared to the temporal pole, and very rapid cycle by cycle traveling speeds. And then propagation direction was not consistently modulated by behavior or the trial content, but we did see 2 consistent and distinct ripple sources along the hippocampal body on average that did have some links to behavior. But we would need to study this a lot more with higher density recordings, higher density grids, ideally across the whole axis of the hippocampus if possible. And also simultaneous laminar recordings are needed as well. So just give a quick shout out to my lab, especially to Monica Chow, who did the bulk of the ripple work. Is a really talented neurophysiologist, but also a clinician. And yeah, there's just a QR code for my lab, and we're always looking for interested trainees, postdocs, et cetera. So thank you.
John, that was wonderful. Thank you so much.
Great, thanks.
I really appreciate it, and I appreciate it especially not only the detailed analyses, but also the higher level introduction you gave about the hippocampal anatomy, as well as the phase stuff, was really, I think, helpful for the whole audience. So thanks a lot.
Yeah, I like to talk about it. It's just, I remember first understanding that stuff back in grad school, and I was just like, wow, this is the most amazing stuff. Like, like so many things can be explained by waves, and just understanding the properties of just a simple Fourier transform is well worth the effort to get that if people are just starting out in this field. So, yeah, I know I went over those, so
Awesome.
hopefully we have some time for
Yeah,
questions.
I think we have some time. Uma and Irfan, do we have questions in the Q&A? I don't see any, but maybe that's a technical thing.
I don't see any in the Q&A, but I had a quick question, if I
Yeah.
could kind of start off. I was wondering if you could talk a little bit about the interaction between these low frequency bidirectional traveling waves and the traveling ripples that you're now looking at.
Mm. Yeah, great question. So, one thing we have not done is to try to link between the low frequency waves, which we have all the data and all the results, and the ripples that we have. Again, have all this data right there. So we haven't looked at it quantitatively, but as you can see from the data, especially during behavior, the low frequency waves were propagating really consistently, especially that 1.9 hertz one, toward the temporal pole. And during this whole like sort of naming process, whereas the ripples that we were recording in the same trials were happening much more often in that initial maybe half second to a full second right after basically the same time frame.
Mm.
So both happening at the same time, what's the deal, right? Like should they be mutually exclusive or not? Again, well, we often think of like theta versus ripples as sort of somewhat mutually exclusive in rats. But in humans, this isn't theta we're talking about. We're talking about like a 1.9 hertz, a 13.8 hertz rhythm. So what I'd say to that is, we need a lot more analysis on this. We need more patients. There's only 6 patients from this little sort of unique but precious data set that we have recorded several years ago. Haven't been successfully at getting larger grant funding to get more data yet. But that's one thing I would love to look at, is to put this together and say, can you predict why, why ripples would happen over top of like low frequency waves? My hunch is that basically they're serving independent functions. So the lowest frequency wave, as you saw, like we had a 1.9 hertz and a 13.8 hertz in the same patient. And then during the intertrial interval, both of those same frequencies, they were headed up toward the septal pole. But during the task, only the 1.9 hertz one was headed toward the temporal pole. I think that's largely reflective of the hippocampal cortical interactions that are happening at that given times. And to be able to explain which exact structures are interacting, I would love to do that. We can only really kind of speculate which structures. But again, ripples are sort of an intrinsic phenomenon. And my thought for them being so local is that they're happening in those local areas because of the cortical connections that are happening at that site as well. So not only are the cortical hippocampal connections driving sort of the low frequency pattern, but over top of that, you're getting local computations happening. So um,
Right.
essentially, lower frequencies are sort of driving the ship in terms of what's happening and like the sort of phase timing of the hippocampus, but the ripples are happening over top of that for local computations that are probably sent out thereafter.
Very cool.
Long-winded answer to your question, but as you can tell, it's a lot of speculation. This is why we need more study and more answers. So,
Yeah. That was a really helpful thought process. We do have a bunch of questions
mhm.
coming in the chat now.
Cool, nice.
Um, so we'll have people come up and um, unmute and ask their questions to you directly if they'd like to.
Sure.
So
Yeah.
first, we'll take a question from Subhashini Lakshmanan.
Hi, yeah, thank you. It was a nice, interesting talk. Can you hear me?
Yep. Can you hear just fine? Yeah.
Yeah. So my first question is the neuronal recordings that you had for non-behavioral tasks. So were they spontaneous, like they were not stimulus evoked? Is that?
Oh yeah, totally. Yeah, I mean, we just did the task for a set amount of time, but then the rest of it was just free recording while the patient was just sort of waiting between neuropsychological tasks and things. And sometimes they were talking to the people in the OR, me in the OR. I was like bringing up a machine there, and so it was just kind of natural sitting behavior for the bulk of these ripples that we got. But the later analysis, which I showed for behavior, those are the behaviorally focused ripples. So
And there you had used visual stimulus, right?
sorry,
For the
say again.
behavioral task, you had used visual stimulus, right?
Yes, exactly. Pictures of like a dog or like other objects essentially, and they were adapted from the Boston naming task. We just basically took, I think it was Boston naming. We took a couple of the Boston naming task stimuli. So
So
Yeah.
with this, having said that it was spontaneous activity, somewhat similar to spontaneous activity, were the subjects of similar age group? Because we did,
Mhm.
I mean, there are certain articles where you can see changes in spontaneous activity for mice, in mice across different ages in
Yeah.
auditory cortex. So,
Oh, that's a great question. Yeah, boy, I wish we had like 100 or 1,000 recordings like this across the age ranges because I would love to look at
mhm,
those parameters. And of course, also across the disease spectrum. As I mentioned, the person with the worst severe or the most severe hippocampal sclerosis had unidirectional waves only instead of bidirectional, suggesting that maybe part of that circuit sort of broke down. But in terms of ripples, I mean, we just these two awake patients, which were the analysis that we did for the ripples, they were, I think, approximately the same age, both in their 30s, I believe, or 30s or 40s. And then the rest of
mhm,
the patients where, we did more of the low frequency analysis and under anesthesia, those patients were also, I mean, it's between 20 to 50, I think was our age range across the six patients. So we're very undersampled in terms of our total number of patients. So I think the
mhm,
the bigger strength of this data set is just having 32 sites equally spaced on the hippocampal surface right adjacent to CA1, but analyzing bigger questions like that are super crucial is going to be, it's going to be have to be a subject of prior work or maybe like depth. Of course, but depth electrodes are the best to examine that because there's just so many more patients that get hippocampal depth recordings.
mhm. Understand. And so, yeah, talking about the ripples, what is the physiological relevance of it? I was just wondering, like, when it comes
Oh,
to
ripples?
ripples, like, yeah, what would be the physiological relevance, or
Yeah.
in any, yeah, yeah.
Yeah, great question. So Ripple, they've been shown in rats to be really important for sort of memory consolidation. If you have single unit recordings at the same time, you can actually see sequences of neurons firing that replicate what had just, or recreate even what just happened with the rat. If they moved through a couple of different areas of a room and neurons fired in
Mhm,
sequence, they'll fire in the reverse sequence after that, or sometimes the forward sequence before they're about to do the same thing again. So they're very important, we think, for memory consolidation, and also in humans, they've been even shown to happen right before speech onset, or when you show a picture like I showed here, and that
mhm,
suggested that they're also important for dynamic memory processing of bursts of activity. They're sort of representing maybe packets of information as the hippocampus is processing information that's coming in, and also information that is also being conjured up from remote memory to reproduce. We didn't have much recall data here, free recall data, but that's a very reliable thing that we see in the literature across many studies showing that ripple rates increase right before someone spontaneously recalls a memory that they had laid down previously.
So, is that something that we can say that ripples is a property of like memory, like it's associated with more with memory? Is that why with the electrode where you had the channel that was outside the hippocampus had less number of ripples?
Oh yeah, and for that I was mainly just yeah, so it's an intrinsic property of the hippocampus, and the
Mm-hmm.
electrode that was basically on the edge. I think you're talking about the where I showed the number of
The
ripples per
bottom right.
electrode. Yeah, the
Yeah,
number of
yeah.
peaking ripples
Uh-huh.
per
Uh-huh.
electrode.
Yeah, yeah.
One was just sort of on the lower right corner, and that's the one that
Mm-hmm.
was it was still on the hippocampus, but sort of maybe just slightly off. Who knows? But also since it was like not, or since it was just maybe even slightly off or only half on the hippocampus at that point, that might be why the number of ripple peaks were detected there, because the amplitude itself would be lower just naturally by having the electrode not fully on. This is speculation again because, and I'd have to go back to the original recording, the pictures and the video that we took intraoperatively to see, but it's just my speculation why, that particular electrode had a lower amount of ripples. It was maybe just kind of only half on the hippocampus.
Okay. Thank you.
Oh, great questions. Thanks.
Cool. We have just two more questions in the chat and then we'll wrap up. There's a question from Brad Caldwell. Unmute
Yeah, it looks
yourself
like
and ask directly.
he's there still. I don't know. He maybe. Oh yeah, I think he's there.
Hello, can you hear me?
Hey,
Yeah,
Brad. Yep.
great talk. I appreciate it. And just a couple of questions. Was the waves when you saw delta and alpha at the same time traveling waves? Were they the same direction?
Oh, great question. Definitely. So we even tried to analyze this and see if we could predict one frequency's direction based on another frequency's direction, and we basically couldn't. There was really like little to no consistency for one frequency versus another frequency in terms of the way that they traveled from patient to patient. Now, that being said, in behavior, I did show the example where during the intertrial interval, the two frequencies present in that patient, so 1.9 and 13.8, did travel more septally or towards the septal pole in the intertrial interval. But during the task, it was only really the 1.9 hertz frequency that consistently traveled toward the temporal pole. And the other frequency, like the 13.8, could have been traveling in either direction or even some other sort of veered off directions. So the short answer is we didn't see really consistent co-traveling waves in the same patient. And again, I think that's reflective that each of these frequencies is sort of has a physiological role that may be independent of the other frequency, but it also allows dual processing to happen. So what if, imagine that 1.9 hertz frequency is driven by interactions with the anterior temporal lobe cortex versus the 13.8 is driven by the interactions with the parietal cortex. Both of them could be processing at the same time. Those oscillations could be traveling in completely different directions, but they're both driving hippocampal physiology. And again, we speculate, at least in the paper, and I still think that this could be one of the mechanisms by which the hippocampus is able to combine information together. So having like cell firing primed and locked to two different frequencies, which are driven by two different neural processes. That could be a way to sort of have both of those processes reflected for ultimate memory consolidation.
Okay, very interesting. And if you had time for another one, it was just I like the idea of the ripples and how you said you get a little blip of ripples right before you basically recall a memory.
Mhm.
And I think a lot of like consciousness might have to do with like frames like that, where you get a little blip and then it kind of expands out into something. But it seems like other regions can do that too. Maybe not with
Mhm.
ripples, but like basically you could, if you're eating chips or something, you could have a little flick of, hey, what if you bit your finger, and that'll make you be more cautious of like. So it's almost like these
Mhm.
imaginative little bursts. And I don't know that that would be hippocampus. It seems like it might be a different brain region, but do you have anything
Hmm.
along those lines, or is that just kind of
So you're saying that basically like a lot of structures could also have increases in activity before speech onset? Is that what you're
well,
saying?
I'm wondering if like everything in consciousness could start with frames and these little blips, and they kind of like expand out into more fleshed out meanings. Like even your understanding of reality, your model of the world could be a series of these little blips, not from hippocampus in that case, but like the hippocampus would be injecting a frame to remind you of some so and so memory that's pertinent to the
Hmm.
situation at hand. And like you have like common sense where some other system interrupts to say, hey, what if this happened? And that
Yeah.
prevents you from doing something stupid.
Right, right. So, what's the mechanism or role of these in, that just recalling a word? So just kind of parsing it down to this example only, is the ripple causative? Is the fact that it happened sort of igniting this memory representation that is then fed to the cortical networks so that the person can produce the word? Or is the word already, produced? It's coming into mind, meaning like different brain regions, and then the hippocampus has a ripple, as a little blip, as sort of an epiphenomenon, right? Meaning like just it's reflected there, but it's not really necessary. So we don't know the answer to that. I would love to see or someday even do that study where we can sort of disrupt these, similar to the work that like Lauren Frank's lab has done, where they sort of detect ripples as soon as they're about to happen, you sort of zap them away and to see if the person has trouble, recalling the word. But I think that their role is essentially in, if not at least reigniting sort of the memory representation, it may be also in sort of sending that memory representation to the rest of the cortex to complete the task. Again, that's just speculation, but that's where we're headed in the field, and that's where we have to, we have to know these answers in the future.
Okay, thank you.
Mhm. I think
Thank you.
Sage Chen.
Yeah, we have one more question in the chat.
Yeah, so
Hey,
can you hear me?
yep, can hear you.
Hi, thank you for the excellent talk. Really enjoyed it. So I have a
Oh,
couple
thanks.
questions. Some of them could be just like curiosity, could be probably you know the answer. So do you see any link between the number of the ripple or the ripple frequency respect to the recall performance for those away subject?
So you mean the peak frequency of the
Or
ripple?
any statistic at all related to ripple respect to the is there any correlation respect to the recall?
The recall. And you're just talking about free recall and not just the naming?
Free recall.
Yeah, unfortunately, we only had like 6 or 7 trials of free recall. So, I mean, as
Okay.
you saw, like kind of squeaked by with getting out of the cluster-based permutation test for right before speech onset, which is the most likely time that we would expect it to increase in terms of ripple rate. But we really just looked at ripple rates and did not look at finer grain details related to behavior, such as the peak frequency, which is kind of going to be corralled in there anyway because
Okay.
of the way we filtered and any study, the way you filter it. But I would love to look at that, particularly for free recall, because a lot of work has been done on that. It's just to do free recall, you have to have the patient do a certain number of trials of a task or give a certain number of stimuli, like a list, and then you have to have the recall done later. So just naturally, we're going to have less recall trials than our standard task, which is visual naming. But that's definitely next up for whenever we can restart these recordings someday. We'd want to do them with a lot higher density of grids, which we can do now, like less, we can do like 0.5 millimeter spacing with our from our collaborators at Lawrence Livermore. Or if other people are producing grids like this, they're literally just the hippocampus is right in front of you, and I'm making it sound way easier than it actually is, of course. But you can put it right over the surface and get many data points recorded. And anyway, so short answer is we haven't looked at it in detail for free recall just because of shortage of trials, but can't wait to do that someday.
Yeah, so another question I have is that you don't see, I'm wondering whether, you see this the bidirectional traveling wave pattern compared to the rodent recording, they have this electrophysiology
Yeah,
recording. So I actually have a conversation with Buzagi, I'm from
hmm
NYU. So
yeah,
he speculated. I mean, obviously, it's also speculated. He said whether it's
hmm
possible is that you see this patterns because it's on the hippocampal surface rather than it's kind of within the hippocampus.
mm,
Um, do you
yeah.
think there's any way we can sort of resolve that sort of ambiguities of whether this is because the surface or is because of
hmm
something? Is any way we can figure out the
Yeah,
differences?
that's a great question. Really a great question, actually. So, Um, so first of all, I'd say that our grid recordings, I kind of showed this in the earlier part of the talk, and really to underscore it, basically they're over the CA1. And as you go from the pyramidal, and this was shown in Bujaki's study and in the Lubinov study, they both showed that when you go from the pyramidal layer, the pyramidal cell layer, north, meaning like superior, or basically towards the hippocampal surface, from the pyramidal layer to the hippocampal surface, there's almost no phase difference. So if you have electrodes along the hippocampal surface, at least we figure that it's probably sampling what the pyramidal layer is doing at that moment in time.
Okay.
And we don't have any electrodes in past that, which would then give you like an offset, a phase offset of some sort, which is going to give you noise because you don't know exactly how far you're going to be. And so anyway, having these homogeneously over this hippocampal CA1 surface is a good way to, we feel a lot more sure that we're recording consistent hippocampal activity from the CA1 in phase. So then whether the bidirectionality is due to the surface recordings, I don't think so, because I think it's actually more likely because of, well, there's two reasons I think this could be. One could be the position. So the Lubinov study was done predominantly on the dorsal hippocampus and a bit on the intermediate. And the Bujaki study mostly again focused on the dorsal, but they also sampled a bit of the intermediate and the ventral. So the Bujaki study was really sort of more comprehensive in sampling the full axis, right? Our study is sampling from, but again, even that study had limited sampling of the ventral and intermediate portions relative to the dorsal. Now our study is predominantly sampling the ventral and intermediate. So we're basically looking at an area of the hippocampus that had a lot less data for the animal studies. So it
Okay.
could be just where we're recording. It's possible, again,
Yeah.
speculation, but if we could record from many sites in the animal ventral hippocampus, maybe we'd see bidirectional traveling waves. It's just that, they didn't have as high a density of electrodes there. And in converse, if we were able to sample from a longer length of the hippocampus, we might be able to pick up on, we could parse out and say, do waves, if we just sample from the dorsal part, to travel anteriorly or posteriorly more often. I did show one example of that where we moved a square grid across the anterior to the intermediate portion. And again, there was bidirectionality along the whole axis there. So that didn't change. So my speculation is that it could be that where we're recording, and the alternate explanation could be it's because it's in rats versus humans. Maybe humans have circuitry that is now able to establish sort of bidirectionality. That may be, who knows? I mean, if we're really going to speculate here, could that be a reason why humans are able to accomplish a lot more in terms of like higher level cognitive functions? I don't know.
I remember this one paper published also in the, I forgot which journal it is. They talk about traveling wave pattern in this epilepsy patient. They have this IED pattern. They describe it as a traveling wave as well. Do you see anything in your epilepsy patient? Like during this intericital epileptic full discharge event, because I wonder if this thing happen in your.
Yeah, totally. So I was gonna maybe include an extra slide or two on this, but I didn't. Sorry, but yeah, for sure. So 5 of the 6 patients have interictal discharges, and we have really nice interictal discharges for many of them. There's actually phase offset for these interictal bursts. We don't have ictal data, so there's no seizures that happen during these recordings, but some of these were like really consistent spikes. And then we, of course, had to exclude those for all the analyses that you saw in here. I personally looked through all the data, found any kind of sharp artifacts, got rid of all that, and we only used the leftover data. One patient had a normal hippocampus, presumably because there was no epilepsy in that patient. But short answer is we definitely have that data. That's another thing I want to look at. So again, if there's anybody that's interested in traveling wave work, especially like applied to the clinical aspects here, it's an ongoing thing that we want to be analyzing more of in the future, even in the same data set.
Thank you very much.
Yeah, great questions.
John, we were having a little text message conversation among me and the other organizers, and I am allowed to ask the last question of you.
Oh, nice, cool.
So,
oh man, it's gonna be a terrible one.
This is a great talk. No, it's not a hard question. It's a fun question, and I really enjoyed the talk because you managed to connect 2 areas of things that I think are really interesting: ripples and also lower frequency traveling waves. So I find it very thought provoking, and I might follow up with some stuff later. But my question for you is about ripples, and you had a couple of plots in the bottom right corner of the slide about the spatial, basically about the spatial scale of visual ripples. And I thought that was really interesting because one thing that we've seen in humans compared with rat ripples is that generally human ripples are lower in amplitude. But what I think you are showing is that if you can measure ripples all over the place, you can find, this is how I interpret your results, so tell me if you agree. One can find the location with these high density recordings. You can find a spot where the ripples are really big in amplitude, and then they have a spatial scale of spreading out. So really, it's not that human ripples are lower in amplitude than in rats. It's more just that they're more spatially confined. So if you don't have those high resolution recordings, then maybe you measure them at the lower amplitude part of them. Or, does that make sense?
Yeah, it does make sense. It makes full sense. Yeah. So, there's a great study, again, by the Bijaki Lab. I can't remember the first author. Oh, it's a while back.
There's a lot of them.
We cited in our paper, though, and showing that they mapped out a single CA3 neuron. This is back in like the late 2000s. A single CA3 neuron and its connectivity along the hippocampal axis. It was connected to like 2/3 of the entire hippocampal axis in a rat with like 40,000 synaptic boutons. So that's a huge amount of connectivity for one structure, right? Now, are we sure that like the hippocampus scaled that way in humans? Is it that interconnected across the entire axis? I'm not sure. That would be kind of surprising in a way, though, because we're talking about like a 4, 5, 6 potential millimeter or 6 centimeter structure in humans. And so I think it's more likely that the ripple scale should be spatially confined. It would be a bit surprising if the role of ripples was to sort of connect the modalities that are happening across the entire hippocampal axis. But if the mechanism is more to sort of ignite or recreate a pattern that can be local in circuitry and just send that out to a few select cortical structures, then it would make sense that they're spatially confined. But essentially every ripple that we recorded was a spatial scale of about a square centimeter or so. And that's a loose, when I say every, but basically the vast majority of these were within like 1 centimeter of space along the hippocampal surface, which is surprisingly small.
Yeah, you might consider changing that plot to be like half width at half mask, or whatever they I'm tripping over my words now,
Yeah.
but you know what I'm trying to say.
Yeah, exactly. Like a different way to, because we just looked at the max Z score, and then that's going to be confounded a bit by, if you try to look at a ring around it, we only have a 4x8, so you can only accomplish a ring around it for the middle electrodes there, and then going to the next ring around might be even tougher. So
Yeah, very interesting. All right, thank you.
yeah,
Uh,
cool, no problem.
Irfan and Uma, should we wrap up? Okay. John, that was great. To those of you in the
Thanks.
audience, we're going to post this recording with a couple edits online. John, thanks so much. And please come see us again on May or early May, May 5th or something, when Michael Breakspear is talking. Take care, everybody.
Thanks for having me. It's a real honor.
That was great, John. Thanks again. Take care.
Thank you. Bye.